Now today we'll be continuing these PKPD parameters of dosing and frequency of antibiotic. Since we have known that three factors are main important for dose and frequency of antibiotic. The first one is half-life of the drug, half-life of that antibiotic. Second is PKPD parameter, and third is post-antibiotic effect. I have already discussed about PKPD parameters that Cmax over MIC is single high dose, less frequency. Main concept of Cmax over MIC is high dose and less frequency. Time of the MIC is frequently we have to give, and we have to maintain the concentration above the MIC. How much time we are going to maintain, that much is the killing effect. This is concentration dependent, this is time dependent. And AUC API MIC is flexible, it is between that two, but it means that we can adjust the dose. This is single daily dose, this is more frequent dose, this is intermediate dose can be given between that. Now another important role, another important player is post antibiotic effect in determining the dose of and frequency of antibiotic. Since post antibiotic effect means the effect of antibiotic on bacteria after brief exposure to antibiotic when there is a persistent suppression of the growth of bacteria is known as the post antibiotic effect. When we are going to talk about the organism, normally post antibiotic effect is for gram positive and gram negative as well. So this is gram positive, gram negative. Now in gram positive, all antibiotic have post antibiotic effect for gram positive organism, that is beta lactam less than 2 hours, vancomycin 5 hours, then aminoglycoside, fluoroquinolones, then tetracycline, clindamycin. These all mainly they have 2 to 6 hours. So for gram positive organism, every antibiotic have some post antibiotic effect and they vary according to the class. But the major concern is gram negative one, and in the gram negative beta lactam antibiotic have no post antibiotic effect. No post antibiotic effect. That is important. So if there is no any post antibiotic effect of beta lactam antibiotic, that is penicillin, cephalosporin, monobactams, exception is carbapenem here. Carbapenems, mainly imipenem, has post antibiotic effect against Enterobacteriaceae and Pseudomonas. So what we are saying that for gram positive organism we have post antibiotic effect of all antibiotics, but when it comes to the gram negative organism, then post antibiotic effect of beta lactam antibiotic is not there. So any antibiotic that have no post antibiotic effect means its drug concentration should be continuously maintained in above the MIC for killing the organism. So from now you have to understand that if there is a GNV organism, GNV sepsis, GNV infection, gram negative organism, what I am talking about, this is gram negative bacilli or gram negative organism, then you have to understand that if you are choosing the beta lactam antibiotic, Then previous PKPD parameter also suggests the same thing. Since it has no post-antibiotic effect, then its dose should be given frequently. That you have to understand. Okay, exception is carbapenem. Carbapenem, mainly imipenem, has post-antibiotic effects and it usually lasts for 2 to 4 hours. So when you compare that, if you are giving imipenem and you are giving 6 hourly, 500 mg 6 hourly. So there is 6 hour gap. Imipenem, if you give 2 hour infusion, so MIC is maintained till 2 hours according to PKPD parameter. Even its concentration has now gone, then 2 to 4 hours it will have post-antibiotic effect. Since it means that if you are giving imipenem to the critically ill patient and you are giving over prolonged infusion, that is 2 hours, then 2 hours it covers, it maintains over the MIC, and the remaining time between 2, this is 1, 2, 3, 4, that is 6 hourly. Now 2 hours is above MIC and the remaining time will be covered by post-antibiotic effect. So whenever look antibiotic, it seems easy, everybody uses it, okay, but you have to understand that there is always need of an expert consultation when there is a serious infection. When you are not able to control any infection, do contact the infectious disease specialist. If you have a heart attack, then you are a general physician, I am MD, internal medicine, can manage, but you usually prefer to go to cardiologist. Same that it can be infection gets eases up, it can be managed by any doctor, it may be physician, it may be internal medicine, anyone, but everywhere expert advice is always preferred if you are not able to manage or if you are getting any complication in managing infection. Please keep that in mind. So we have discussed about that gram-negative organism post-antibiotic effect. Beta-lactam antibiotic is not there. So if there is no post-antibiotic effect, the prolonged infusion is always preferred. Now if we are going to talk about other group of organism, then other group of antibiotic, then aminoglycoside and fluoroquinolones. These have 2 to 6 hours of post-antibiotic effect. You have to remember one thing, that this aminoglycoside acts on the protein synthesis inhibitor. Fluoroquine acts inhibit the DNA kinase. So these are acting on the protein and nucleic material. A bacteria takes longer time to develop this protein synthesis and nucleic material in comparison to cell wall. Because of that also post-antibiotic effect is seen in bacteria. Okay, then there will be tetracycline, clindamycin. These all have post-antibiotic effect against GNB. Okay. Now, important factor is that when we are talking about two things, that is, let me talk about polymyxin. Polymyxin B has found that it has post-antibiotic effect against Pseudomonas, but no post-antibiotic effect against Acinetobacter and Klebsiella pneumoniae. So what does it mean? That it means that whenever you are going to use polymyxin, use always in combination with other antibiotic if you are going to treat Acinetobacter and Klebsiella. Why is that? Because since it has no post-antibiotic effect against Acinetobacter pneumoniae and Klebsiella pneumoniae, the problem is that when you are using polymyxin B for say 7.5 lakh BD or say 10 lakh BD, then you are, since it is not having post-antibiotic effects against Acinetobacter and Klebsiella, then when the antibiotic level goes down, there is a heteroresistance occur and it regrowth of this organism occur. Even resistant can happen. Sometimes it has found that TDS in murine model, in animal model, when we have used this polymyxin 3 times, divided the dose into 3 times, then have found to be suppressing this heteroresistant and prevent from occurring the resistance of the organism. That is also one of the point that I think it is important to mention here. So polymyxin dose is usually we give PD, but it can be given TDS on the basis of as it has parameter of AUC by MIC, so it is flexible, okay. And also since it has no post antibiotic effect against Acinetobacter and Klebsiella, so its dose can be from BD to TDS or used in combination with other antibiotics. That is to be measured. Okay. Now talking about the aminoglycoside. Aminoglycosides, it is Cmax over MIC. So single dose reduced will be concentration dependent kinetics and killing. Second is post antibiotic effect for both gram positive and gram negative organism. So it is better. Three is post leukocytic enhancement. Enhancement is there for killing of the bacteria. But important thing is it doesn't work in acidic pH. So in abscess there is acidic pH and less work, and so no aminoglycoside. It doesn't mean one thing that aminoglycoside in acidic pH, that wherever the abscess is present there is a metabolism and acidic pH is maintained there, so it doesn't work properly. That is one major thing of aminoglycoside that you have to know. Other you know that it has Cmax by MIC post antibiotic effect and post leukocytic enhancement is also there after this using antibiotic. Okay, so hope you have understood that for determining the dosing and frequency, three things comes a key role play. That is, one is half-life of the drug, second is PKPD parameter, and third is post antibiotic effect. Hope this are helpful.